Hey, this is Mr. W, and this video is about the specific immune response. We're going to learn about some amazing stuff, like how your body can produce proteins called antibodies that can neutralize viruses. That's what's going on in the image to my left. This video is the second in a two-part series. In the first video, we discussed how the immune system has three lines of defense. We focused on the outer barriers, the skin and the mucous membranes, and the nonspecific or innate responses. In this video, we'll look at the third line of defense, the specific responses. It's also known as acquired immunity because you acquire this type of immunity over the course of your lifetime. It involves two cell types, B cells and T cells, and there are three steps: recognition, overcoming, and remembering. This video is linked to a series of 3 interactive tutorials on Sciencemusicvideos.com. The link is below. Watch this video and complete those tutorials, and you'll have a sophisticated understanding of how immunity works. Also, I have a song about the specific responses, but you don't have to go anywhere to see that. I'll embed it into the end of this video. Let's start with an evolutionary view. What do all of these organisms have in common? The reason why this is important is that while all organisms, even bacteria, have some type of system for fighting off infection, only vertebrates have the type of specific responses that we'll be talking about in this lesson. The immune system is based on 2 cell types. These cells are called lymphocytes because they're often found in lymph nodes, which I'll talk about more in a minute. The first lymphocytes to know about are B cells. These cells mature in the bone marrow, shown at 4 in this diagram, and their function is to secrete antibodies, proteins that fight off viruses and bacteria. T cells are the second type of lymphocyte. They mature in a gland called the thymus, shown at number 5 in this diagram. Upon maturation, they become helper T cells, the cellular generals in the immune system army, or killer T cells, cellular assassins that can find virus-infected cells and destroy them before they can make more viruses. So what is lymph and what are lymph nodes? Lymph is a clear fluid that drains from body tissues. If you took blood and removed the cells, the clot-forming platelets, and some of the proteins, you'd have lymph. In other words, it's similar to the part of blood that's called plasma. Lymph nodes are where lymphocytes live, the B and T cells, and it's where they spend most of their time. The nodes are where much of the action involved in the specific immune response happens. When you're fighting off an infection and you have swollen glands, what you really have are swollen lymph nodes. It's a sign that your immune system is ramping up. The term antigen is one of the most important terms in understanding the specific immune response. It's a relatively new word, invented about 100 years ago as scientists were figuring out how immunity worked. It means antibody generator. It's what the immune system recognizes and responds to. It's not an entire pathogen, but a piece of the molecule from the pathogen, usually a fragment of a protein or a polysaccharide. In this diagram, the different shapes indicated by the arrows at 1 all represent antigens. In reality, they would have a very complex 3-dimensional shape. You can see that in this image. On the surface of SARS-CoV-2, the virus that causes COVID-19, is a spike protein. It's actually a trimer, a complex of 3 proteins. That spike is possibly something that could be recognized by the immune system as it mounts its counterattack. In other words, that spike is an antigen, an antibody generator. So what are antibodies? Antibodies are proteins secreted by B cells that bind with and neutralize invading pathogens. Here you see an image of B cells at 1 producing antibodies, at 2 that bind with a virus. Once bound with antibodies, that virus can no longer bind with cells, and if it can't do that, it can no longer infect cells. Antigen testing is a way of detecting the virus. An antigen test for SARS-CoV-2 would involve seeing if a person had in their blood or other tissues some piece of that virus. That could tell you if you were infected. Antibody testing would test your blood to see if you had antibodies to the virus. That could mean that you were immune to the virus. I'll explain more about that later. The way that your immune system recognizes antigens on invading pathogens is through receptors that are on the surface of B and T lymphocytes. These are represented by number 1 in this diagram. It's all about the shape at the tip of the receptor. So let's take a closer look at how these receptors are made up. Here's a very important concept. B cells, as part of the immune response, secrete their receptors into the blood. These secreted receptors are antibodies. So when we talk about antibody specificity, we're also talking about receptor specificity. Also, I want to point out that what I'm saying about B cell receptors also applies in large part to T cell receptors. Let's start with the B cell receptor. In this diagram, number 1 represents the B cell. The entire structure at 2 is the receptor. And just so that you know, this is completely not drawn to scale. The antibody is a quaternary protein made of 2 pairs of polypeptide chains. The longer chains at 3 are called heavy chains. The smaller chains at 4 are called light chains. The tips at 5 are what binds with the antigen, and it's called the antigen binding site. Now, note the incredibly complex shape of this antibody, especially at the tip of the arms. That shape is the key to recognizing and responding to pathogens. It's the antigen binding site, and that leads to a mystery. We have about 20,000 genes in our genome, but humans can generate billions or even trillions of specific antibodies, each of them a unique protein with a unique amino acid sequence and therefore a unique shape. How do we do that? The details of this could be a lesson in and of itself, and I want to encourage you to read my written explanation on Sciencemusicvideos.com. But here's the basic idea. The genes for receptors, and therefore antibodies, consist of many different exons, separated by introns. If you need a review of introns and exons, I have a separate tutorial about that, and I'll link it below. As B and T cells develop, the exons are randomly shuffled around. This is shuffling at the DNA level. This shuffling is like the shuffling of playing cards. I have a friend who's explained to me several times that every shuffle of a deck of cards creates an enormous number of possible decks, 8 times 10 to the 67th different combinations. So exon shuffling similarly creates enormous antibody diversity. On top of that, there's a high mutation rate in antibody genes that occurs as B cells develop. That further enhances receptor and antibody diversity. We can create enormous varieties of B and T cells, as I just described, each with a unique receptor. But each one of us is a finite being, only so big with room for only so many cells, though it's a huge number. For your specific immune response to be unleashed, there has to be a B cell with a receptor that can bind to the antigen on a pathogen. That's what recognition is. Here you see 3 B cells, each with a different receptor. The second one has a receptor that matches the antigen, so we're in good shape. We've recognized the antigen and we can unleash our response. But there's not always a cell number 2 or its equivalent. The reason why there's a pandemic now is that this coronavirus, SARS-CoV-2, is a novel coronavirus. It has antigen that we haven't seen before as a species. And as a result, many of us aren't responding well, so a lot of people are getting sick. This is what epidemics and pandemics are all about. If there's no B or T cell with a matching receptor, you can't respond very well. And that leads to one of the biggest misunderstandings that people have about these receptors and about the immune system as a whole. That misunderstanding is often stated like this. When I get infected by a pathogen, my body responds by creating custom-made antibodies that fight that pathogen off. But it's not like that. Let me talk about the way it is by way of an analogy. How I buy my pants. If I were a king or a lord or a billionaire, I might have my clothes custom made. A tailor would come to my house and make a pair of pants just for me. But I have never bought a pair of pants like that. Instead, I go to a store and find a part of a room that looks like this. The pants that I'm going to wear are waiting for me. And that's how it is with the immune system. As my immune system was developing, all sorts of receptors with unique shapes were created. Now they're sitting in my lymph nodes waiting for the antigen that fits them. In other words, antibodies are not tailor-made. Antibody diversity in each of us is pre-existing. Again, why are we in the midst of a pandemic? Let's read this together. If you, as an individual or a population, don't have a receptor/antibody to a pathogen, then unfortunately you're out of luck. All of that was so that we could understand the first step in specific immunity, recognition. Let's move on to the next step, which is overcoming, and then we'll move on to remembering. Overcoming has two parts. On my right, you see a representation of what's called the humoral response. In this response, B cells secrete antibodies into the bloodstream. On my left is a representation of the cell-mediated response. In this response, cells called killer T cells or cytotoxic T cells find cells that have been infected by viruses and wipe these out so that they can't produce any more viruses. In both the humoral and the cell-mediated response, a key process is clonal selection, which in some ways is part of the previous section, recognition. To explain this, I'm going to focus on the humoral response, but the basic idea applies to the cell-mediated response too. Remember when I was talking about buying pants? I walk into the store and the pants are already waiting for me. In the same way, we have a variety of B cells with different receptors. The cells here are shown at 2A, 2B, and 2C. Their receptors are indicated by number 5. These cells are already there in our lymphatic system, waiting for an antigen to bind to them. The antigen is at 1. Note that only cell 2B has a receptor with a shape that complements the shape of this antigen. Because of that matching shape, cell B becomes activated and gets cloned into millions of copies. The process of cloning is indicated by 3, and the resulting clones are shown at 4. Note that the antigen binding site on every cell in this clone is the same. That's the point. They're clones. As the cells within this clone continue to develop and differentiate, they have two basic fates. One is shown at 7. These cells develop into plasma cells that secrete antibodies. Because of clonal selection, the antibody that's secreted has a shape that enables it to bind with the antigen. As it does, the pathogen that the antigen belongs to will be vanquished, and I'll show you how that happens in a moment. Other members of this clone will develop into memory cells, shown at 6. These memory cells are ready to spring into action at the first sign of the next infection by the same pathogen, and that is what being immune is all about. To review, in terms of the overcoming part of the humoral response, this is the result of clonal selection, a clone of plasma cells that churn out antibodies that are targeted against a specific antigen. So what we have to do now is explain how in the vertebrate body this actually works. This is going to involve connecting some things we learned about in the previous video, non-specific innate immunity, with the specific response. Remember these phagocytic cells, macrophages and neutrophils? They're roaming the tissues, finding invaders and devouring them as part of the non-specific immune response. That's shown in step 1. Then these phagocytes digest the pathogen enzymatically, breaking it down. That's step 2. But remember that these phagocytic cells are not only defenders, they're also sentinels. They sound the alarm. Now we're going to see how that happens. These phagocytes display some of those digested pieces of the pathogen in a special protein that's called an MHC class II protein. I like to think of these MHC proteins as skewers. The phagocyte jams some antigen onto the tip of the skewer and then holds that skewer up saying, "Look at what I found." Notice that this MHC class II protein has a different function than the MHC class I protein we met in our discussion of the non-specific response. An MHC class I protein is found on all body cells, and it says to the immune system, "I belong here, I'm part of the team." The MHC class II is found only on antigen-presenting cells, phagocytes. It holds up antigen saying, "Look what doesn't belong here. Let's get to work." How does this actually happen? At this point, a military analogy is useful. The phagocyte was a scout. It now has evidence of an impending invasion. It goes to headquarters. In the immune system, the headquarters is a lymph node. And once in headquarters, the antigen-presenting cell needs to make sure that this message gets through to the rest of the army. This message is going to go all the way to the top, to the general. In the immune system, the general is the helper T cell. It's the cell that tells all the other cells what to do. But at this point, this military analogy breaks down a bit. That's because in the immune system, there are a million potential generals. The helper T cell that gets to be the general is chosen by clonal selection. So imagine this phagocyte standing there holding up the antigen it's found. The first helper T cell comes by, but its receptor doesn't match. It goes away. Then the next helper T cell comes by, and the next, and the next. The process continues until a helper T cell with a matching T cell receptor comes along. That's what we see here. Note that to become the general, the helper T's receptors must be blank to the antigen. What is that? That's right. It's one of the most important words in biology, complementary. Binding of a helper T with an antigen-presenting cell, or an APC for short, activates the helper T cell. The initial bond between the helper T and the APC is a very weak bond. To strengthen that bond, the helper T reaches out with a protein called a CD4 protein. Now these two cells are locked together. That connection is accompanied by the release of cytokines, a type of signaling protein. Some of these signals are from the APC to the helper T. Some of these are autostimulatory signals. Now let's think of this word, cytokine. It should make you think of the word cytokinesis, and that's a clue about what happens next. The helper T clones itself into millions of copies, and these copies wait in lymph nodes until one of them binds with a B cell that has independently found the same antigen and is displaying it. And I know that's confusing, but B cells are also phagocytic, and they can also display antigens. So we're having multiple levels of connection before we activate the system. This leads to another burst of cell signaling and cytokine release. In this case, the cytokines induce the B cell to clone itself into plasma cells. And these plasma cells churn out antibodies. 2,000 antibodies per second per cell. And you have millions of plasma cells doing it. There's an entire clone. So these antibodies are potent weapons against pathogens, and we'll see exactly how in just a minute. But first, let's look at this image, which pulls a lot of this together in cartoon form. Once a helper T cell has been activated by clonal selection, it has to wait to find a B cell that's independently bound to the same antigen. That's essentially another round of clonal selection. Once that's happened, the helper T cell will release cytokines, indicated by H, that induce the B cell to clone itself into plasma cells that release antibodies. But now, let's see what these antibodies do. Because of clonal selection, antibodies bind with antigen on a pathogen. There are three ways in which antibodies help vanquish a pathogen. The first is called opsonization. A pathogen that's coated with antibodies is easier for a phagocytic cell to swallow and destroy. The second is neutralization. When an antibody has coated the surface of a virus, that virus can no longer bind with its target cell's membrane and trick that cell into letting it enter and take over the cell. This is the basis of what's called antibody treatment for COVID-19. Doctors can get antibodies from the blood of people who have been infected with SARS-CoV-2 and then infuse these antibodies into the blood of someone who's suffering from an active infection. The third mechanism involves interaction between antibodies and complement. We discussed complement in the last video. It's an antibacterial group of proteins that's part of non-specific immunity. Here you see antibodies that are bound to an invading bacterium. Those antibodies can then be grabbed by complement, which is shown at letter A. That acts as like a ratchet that blows holes in the walls of bacterial cells, which kills them. Here's the entire humoral response. Right now, here are two good learning moves. The first is to test yourself by clicking pause and seeing if you can talk yourself through every part of this diagram. Second, go on over to Sciencemusicvideos.com and complete the tutorial about the humoral response. The link is below this video. Wow, we have learned so much about the immune system. There are only two parts left. First, we're going to talk about the cell-mediated response, and then we'll turn our attention to immunological memory. Let's go. This account of cell-mediated immunity is somewhat simplified, but it's about right for a survey course at the introductory college or AP Bio level. The cell-mediated response begins with antigen-presenting cells, some type of phagocyte, presenting antigen to helper T cells and killer T cells. Killer Ts are also called cytotoxic T cells. They're cellular assassins. So, as with the humoral response, we're having multiple rounds of clonal selection. This is particularly important here because this response is about attacking our own cells when they've been taken over by viruses or when they become cancerous. Our body only wants to unleash this response when it needs to happen. And even with these multiple checks, there are still mistakes. Misfires of the system can lead to what are called autoimmune disorders, where our own immune system attacks our own organs and tissues. Conditions like type 1 diabetes, lupus, and some types of arthritis are thought to have an autoimmune component. Once the correct helper T and cytotoxic T cells have been identified, cytokines get released from the helper T cell. You can probably guess what happens next. This creates a clone of killer T cells. These cells are released into the bloodstream and then search the body, looking for tissues with a class 1 MHC protein, shown at number 2, that's displaying viral antigen, shown at 1. Imagine these killer T cells roaming the body, touching every body cell they encounter with their T cell receptor, asking themselves, do I feel the antigen I'm looking for? If the answer is no, they move on to the next cell. But when they find a cell with the right antigen, they attack. First, they use a protein called CD8 to grab onto the tissue cell. Then they unleash their chemical weapons. Perforins perforate the membrane of the infected cell. Granzymes induce tissue cell apoptosis, program cell suicide. As a result, these zombie cells that have become virus factories are destroyed. One question my students always ask me about is how this is different from what natural killer cells do. It's actually not that different. The difference is really about the questions that these cells are asking themselves. The natural killer cell is asking, is this cell a part of the body? Does it belong here? Does it have an MHC class 1 protein? The killer T cell is asking whether a cell is displaying viral antigen and therefore become a virus factory that needs to be destroyed. Note how the first question is a lot more general than the second. And that makes sense, since the second question is related to the specific immune response. Now the last step in specific immunity, remembering. Successful overcoming leaves behind memory cells, memory B cells, memory helper T cells, and memory killer T cells, or cytotoxic T cells. We're just going to focus on memory B cells, but the principle is the same for other memory cells. Once you have fought off an infection, you keep a large clone of memory cells in your lymph nodes. These memory cells are specific. They're the result of clonal selection, and they have receptors that complement a specific antigen from a pathogen that you successfully fought off in the past. When that pathogen infects you again, those memory cells are there in your lymph nodes waiting. In the case of memory B cells, they'll immediately clone themselves into plasma cells that will make antibodies that destroy the pathogen. So it's important to understand this. When you're immune, it's not like you can never get infected anymore. You don't have some force field around your body. It's just that if you do get infected, your immune system can fight off the infection before you even know you had it. This graph says it all. The x-axis is time and days. The y-axis is how many antibodies are in your bloodstream. The units are arbitrary. The response shown at B is called primary immunity. It's your initial response to the pathogen. Over the course of a few days, clonal selection enables you to produce antibodies that fight off the pathogen. By day 28 in this graph, you're back to normal. Now, let's say you get infected again. This is secondary immunity, and it's shown at D. You have memory cells, so you produce many more antibodies. The pathogen never has a chance. That's what we're hoping for in those who have recovered from COVID-19. They'll have memory cells. If they get infected again by SARS-CoV-2, we're hoping that they'll be able to fight it off. But note, that's not always the case. I mean, you can get influenza every year. You can get a cold several times a year, and many colds are caused by viruses in the coronavirus family, the family that SARS-CoV-2 is a part of. Vaccination is a way of artificially inducing the formation of memory cells without ever having to suffer from the disease. Today, people forget the toll that was caused by diseases like measles, mumps, polio, tetanus, diphtheria, pertussis, and others. In the pre-vaccination era, it was common for parents to lose their children to childhood diseases. That was the tragic, inescapable reality of life before there were vaccines. Now, these are diseases that almost every American gets vaccinated against. Because of these vaccines, death from childhood infections has become very uncommon. It's one of the big accomplishments of modern medicine. The first vaccines were developed in the 1700s. A virus that caused a mild disease, cowpox, was used to vaccinate people against a deadly, disfiguring disease, smallpox. I was born in 1961, and I'm among the last generation of people to be inoculated against smallpox, a disease that has been completely eradicated from the human population. The word vaccination comes from the word cow, vaca, and it relates to the cowpox origins of this practice. This practice is also called immunization or inoculation. So let's see if you can fill in the blanks here. Vaccination involves injecting harmless antigens so that your body develops memory cells against harmful pathogens. The hope is that we'll be able to develop a vaccine against SARS-CoV-2. That's the best way to get to the other side of this pandemic. Until then, we have to keep our distance from one another, wear masks when we're near other people, wash our hands, and avoid touching our faces. Vaccination isn't just for us and our loved ones. When enough individuals in a population have immunity, then that population develops what's called herd immunity. This varies from virus to virus. For measles, for example, it's about 90%. That means that when 90% of the population is immune to measles, that virus can't successfully replicate itself within that population. When you get vaccinated, you're helping to protect the entire population, including babies and people with weakened immune systems. The last thing I want to teach about related to immunity is the idea of passive immunity. You can develop immunity as you respond to an infection, or you can develop immunity as a result of immunization. Those are both forms of active immunity. You produce the antibodies. So what's passive immunity? That's where you receive antibodies from outside your body. This can happen naturally in any mammal as antibodies pass from the mother to her baby. That can either be through the placenta during pregnancy or through breast milk after pregnancy. Now, the culture around this has changed since I was a baby, when it was unusual for moms to breastfeed. Now about 80% of North American moms breastfeed their babies, and that's great because it helps provide a baby with immunity before they develop their own acquired immunity. Passive immunity can also happen artificially. This can happen by isolating antibodies from the blood of someone who's recovered from a disease, or the antibodies can be made through genetic engineering and then transfused into an infected patient. Both of these are being used, or at least developed, as therapies for COVID-19. Holy cow, that is it. As with my first lecture about nonspecific immunity, I have a song about specific immunity, and here it is. If you've already heard it, please head over to Sciencemusicvideos.com so you can complete the tutorials and really learn this material at a deep, sophisticated level. Welcome to Immune System Wrap Part 2. Now we'll learn what the specific responses can do when innate defenses start to be breached. Antibodies and killer T cells are unleashed. The specific response has three distinct components. Recognize, overcome, remember your opponent. It's acquired, adaptive, and proven over time. It lets you beat back pathogens. It keeps you in your prime. The cells that carry out specific immunity are known as lymphocytes. They go by letters B and T. They're often found in lymph nodes, your body's fluid filters. You feel them get swollen when your system gets off kilter. These specific responses are a vertebrate feature not found in bugs or worms or any backbone-lacking creature. The response is elicited by what's called an antigen, a portion of a molecule on an invading pathogen. Antigen means antibody generator. Polysaccharides are proteins on the cells of an invader. Antibodies are proteins the lymphocytes secrete to bind with unique antigens and lead them to defeat. Both B's and T's can recognize specific pathogens through receptors on their membranes that bind with antigens. A B cell receptor is made of 4 polypeptide chains, which forms a kind of why the stem is stuck stuck into the membrane. Two other chains are heavy, two of the chains are light. The two tips of the Y form the antigen binding sites. Activated B cells secrete their receptors. As antibodies, they are system's #1 protector. Random recombining within B and T receptor genes generates amazing receptor shape diversity. The staggering assortment of B and T receptors lets us recognize specific invaders when they enter. T receptors are quite different, but their function is the same. They combine with almost any antigen that can be named. They're just two chains, and right at their tip is the specific shape that binds invaders in their grip. T cells come in two main varieties. The helper T's, the general in the immune system army, and cytotoxic killer T cells, they're assassins. They kill zombie cells controlled by viral pathogens. It's the immune system, sworn to protect you from dangerous pathogens that try to infect you. 3 layers of defense keep the germs away, letting us live yet another day. Skin and mucous membranes first, then innate defenses for fending off invaders that breach our fences. Then specific responses with lymphocytes B and T. Let's learn about immunity. Let's examine how our B cells respond to an infection. The big picture here is called clonal selection. A B cell receptor binds an antigen with matching shape. This activates the B cell, which divides and makes a clone of plasma cells whose job is to secrete antibodies for that specific antigen they'll meet. Antibody action through our body fluids, it's called humoral. We'll see below the ways it sends invaders to their funerals. B cell activation starts with phagocytic cells, dendritic cells, and macrophages, all these sentinels. Devour a pathogen, then break it down, then hold it up as antigen, show them what they found. The antigen's displayed on a special protein, part of a family called MHC. MHC is for major histocompatibility complex. It's used to say, this is me, I'm on your team, and here's a little something that's trying to infect us. We gotta do something, or this'll be the end of us. Our phagocytic sentinels use MHC class 2, and holding up the antigen, they make their move to lymph nodes where they bind with a helper T, whose receptor is also complementary to the antigen that the phagocytes displaying. This is the connection for which we've been praying. The helper T grabs on with its CD4 protein, which connects antigen presenting cell and helper T, and now the two cells messages exchange, inducing the helper T to change. The messages are in the form of cytokine secreted proteins that are used to define other cells' function and even their form. In this case, they induce a helper T swarm. Helper T clones itself into identical versions, each of which is the receptor for the antigen. These helper Ts wait in a lymph node till they find a B cell to whom the same antigen should bind, which means both B and helper T have receptors that complement the shape of this antigen. Infector cytokines from helper T get B cell to start division, makes a plasma cell army that's devoted to the mission of secreting antibodies 2,000 per second per cell. These bind with antigens on pathogens which feel like hell was unleashed, as antibodies act like a tag so that phagocytes can put those germs inside the bag. It's called opsonization, an antibody trick. Here's another antibody move that's pretty sick. By binding viral proteins, antibodies prevent viruses from binding to cells they might affect. Gumming up these viruses is neutralization, another immune system way of pathogen negation. Antibodies also boost the complement system. Infected cells and germs explode their antibody victims. It's the immune system, sworn to protect you from dangerous pathogens that try to infect you. 3 layers of defense keep the germs away, letting us live yet another day. Skin and mucous membranes first, then innate defenses for fending off invaders that breach our fences. Then specific responses with lymphocytes B and T. Let's learn about immunity. Killer T is an assassin, not to be underrated. The cytotoxic killers do a cell-mediated response. Like B's, killer T's need prior activation by a phagocytic cell that makes a presentation of antigen displayed in an MHC protein, and often helper T's assist this activation scene. Once activated, killer T's clone themselves into a platoon that roams the body to bring about the doom of body cells with cancer or an infection. And here's how killer T's do target detection. Their T cell receptor binds with the antigen, held aloft by tissue cells which are infected. The antigen's held up in their class 1 MHC, the sickly tissue cells, saying, look what's inside of me. So killer T grabs hold of protein CD8 and then proceeds to assassinate the infected cell. It's a mercy killing using perforins for membrane drilling and granzymes to induce apoptosis. Infected cells die from these lethal doses. Remembering's the last step in immunity, allowing you to face repeat invaders with impunity. B and T memory cells are the trick that build up your immunity, keep you from getting sick. Every time your specific responses have a victory, there's not only effector cells, there's also cells of memory. These memory cells stay behind with their receptors, waiting to bind the same disease-causing vector. So in the next infection, you skip all the preliminaries, go right to cloning plasma cells and even killer T's. This mobilization means you'll bring germs to their knees, you'll wipe them out before you feel the symptoms of disease. That's why the first immune response you have is rather about a week. You beat the pathogen, but mobilizing took you weeks. But the second response you have can really be fine. Many, many more antibodies made in much less time. And that's how vaccination works to keep away disease, injecting harmless antigens to develop memory cells with receptors that detect and arm the specific immune response that keeps us from harm. It's the immune system, sworn to protect you from dangerous pathogens that try to infect you. 3 layers of defense keep the germs away, letting us live yet another day. Skin and mucous membranes first, then innate defenses for fending off invaders that breach our fences. Then specific responses with lymphocytes B and T. Let's learn about immunity. Thanks so much. Please subscribe to my channel and head on over to Sciencemusicvideos.com. I'll see you there. Good luck. Stay safe. Thank you.